everyone, I hope you're all doing well. First of all, I wanted to thank the 5 people who watched my last video. Honestly, I wouldn't be surprised if the fame started going to my head, so bear with me if that happens. But in all seriousness, today I wanted to talk to you about virus life cycles. We're going to kind of go through what a virus is, and then we're going to look at 3 types of life cycles that viruses have. So I hope you're excited, and let's get started. So, first off, what is a virus? We hear a lot about it, especially nowadays, but I thought I'd just do a little recap so that we're all clear as to what we're talking about here. So, a virus is a submicroscopic or tiny infectious agent. And what is super particular about viruses is that they cannot replicate outside of a host cell. So they need a host cell to replicate, right? And for that reason, scientists are not sure whether they want to classify viruses as being alive or not, since they cannot replicate on their own. A virus always has a nucleic acid genome, so that's a genome made out of DNA or RNA. It can be single-stranded or it can be double-stranded. And this genome could be cut up into several segments. That's very possible. For example, the virus of the flu, which we are all very familiar of and hopefully getting our vaccines for soon, has its genome cut up into 8 little pieces. Sometimes the viral envelope can include lipids and a membrane, so kind of like in our cells, it just kind of holds their genetic information, all their genome together. And genome complexity actually varies a lot for viruses. The more complex a virus genome is, the less it's going to rely on the host cell's functions, so its proteins and things like that. It's going to be able to take control a lot easier if it's more complicated. Okay, so that was the virus, and now let's move on to the 3 types of life cycles. So the type of life cycle that a virus is going to have is 100% dependent on what kind of genome it has. And here we're starting with a virus that has a DNA genome. Here we can see it's double-stranded DNA. So we are going to call this a DNA virus life cycle. So here's a little virus that I drew in purple with its blue double-stranded DNA inside. And it's going to attack the host cell, which is this big circle in the middle, and it's going to inject its DNA. On one side, so on the left side, you can see that the cell's enzymes are going to replicate the viral DNA, so the blue DNA, just like it would replicate its own DNA. It's not going to notice that it's viral DNA. It just sees DNA and then it knows that its role is to replicate DNA, so it does it. On the other side, you see that the capsid, so this purple membrane or envelope that was surrounding the original virus, is going to be broken down. Meanwhile, some of the viral DNA is going to be transcribed by the cell's enzymes to turn into messenger RNA or mRNA. This messenger RNA actually codes for the capsid proteins that are going to make the envelopes for the new viruses that are going to emerge out of the cell. So on one side, we are replicating the genome, and on the other side, we are kind of making the body that composes the virus. So they're going to come all together, they're going to assemble themselves, and finally form these little new viruses. I've drawn 3 at the bottom there, and they're going to explode out of the cell and go free to go infect more cells. So the cell cycle continues like that. Okay. The second type of virus life cycle I want to talk to you about today is the retrovirus virus life cycle, or the retrovirus life cycle in short. So how is this going to work? You have your little virus with the glycoproteins that are making up the capsid, and these glycoproteins, they help virus to protect themselves from white blood cells or cells from the immune system so that they don't stick to them. So this virus is going to go into endocytosis with the host cell, which just is a fancy word to say that the membranes are going to fuse and the virus is going to be liberated into the cell. So what gets liberated into the cell from the virus? The genome is here a single-stranded RNA piece. It's that little red piece I've drawn for you. That's going to be released into the cell. the capsid and glycoprotein are released into the cell, and finally, a very important or very key factor, the inverse transcriptase. Now, the inverse transcriptase job here, it's viral, is to inversely transcript the virus's RNA to form a DNA-RNA hybrid. So using the RNA as a matrix, it's going to copy the RNA into a complementary strand, but of DNA. So we have this DNA-RNA hybrid, and then the single-stranded piece of DNA is going to be replicated. So it's those two lines in blue that I've shown you. And this piece of DNA, this double-stranded piece of DNA, is going to insert itself into the chromosomal DNA of this host cell. So this is when it's called a provirus, which means it's dormant. This viral DNA is dormant. It's just resting. It can't be detected, and it's going to be transcribed by the host cell into messenger RNA as the host cell would normally do with its DNA. So some of it is going to be used to make genome for the next generation, and the other is going to be transcribed into more protein that's going to form the capsids and the glycoprotein, kind of like we saw in the first one. It's going to make the viral envelope of the virus, and so everything comes back together again. They assemble, and the virus is let out. Alright, so the last type of life cycle I want to talk to you about is the RNA virus negative positive strand life cycle. I put negative strand here because it is the more complicated type, but we're going to go through it. Basically, this type of virus, it has a RNA genome and it's single-stranded, and it's either going to be of type negative or of type positive. So what happens? This, as always, our virus attacks the cell, the genome is inserted inside, so are the capsids, everything is inside the cell. The viral genome, so the RNA, if it is a positive strand, it's going to be directly transcribed into proteins, into the glycoproteins. It's going to be directly copied by viral DNA polymerase, so the polymerase that is the virus's property. It's its own. Everything's going to come together, and the virus is going to eject from the cell. However, if this strand was a negative strand, then it must first be copied into a positive strand. So the complementary of its strand will be positive. So just a normal RNA replication, and it can go through all the functions as the other one. So it's going to be copied, transcribed by the cell. There's going to be a copy of RNA by the viral RNA polymerase. And just so for your information, but I've written it down here, the negative strand is going to be copied by an enzyme, which is the virus's property, called a replicase. So this is the enzyme that's going to turn the negative strand, well, copy the negative strand into its complementary, which is the positive strand. And this process, these copy enzymes, don't really have any proofreading, which means there's often, which is approximately every 10,000 bases, an error which causes mutations in the genome of the virus, which is going to really increase the genetic diversity of these viruses, which is very beneficial for them actually, because they are going to be able to adapt to new environments and acquire new characteristics that will probably help them. It can be deleterious, so it can be bad for them, these mutations, but it can also be very good for them. Alright, so those were the 3 virus life cycles I wanted to show you. I hope you enjoyed this video. As always, if you did, make sure to let me know. Please ask any questions you have in the comments below, and let me know if you want a particular topic to be covered, whether this be in chemistry, math, physics, biology. Alright, bye guys!